The primary response in the context of an infection is the sensitization of the immune system to previously unknown pathogen antigens. In the case of protein antigens, the primary response is initially via MHC2-mediated antigen presentation by dendritic cells, monocytes, macrophages, and B lymphocytes. After contact with the MHC2 receptor of the Th0 cells, T lymphocytes, IL-4 proliferates to form the Th2 cell, and at the same time proliferates via IL-2 to form the Th1 cell. The Th2 cell initiates the proliferation of B lymphocytes via IL-4 to antibody-producing plasma cells. Optionally, existing plasma cells can be directly stimulated to form antibodies by activation using polysaccharide-based pathogen antigens. The specific antigen receptors of the B lymphocytes bind to the pathogen antigens and are taken up as a complex by endocytosis. The receptors are then multiplied and released as antibodies. As a result, pathogens are opsonized by the antibodies formed via antigen binding and marked for macrophages, the complement system and granulocytes. Likewise, the antibodies formed act as a direct activator for pathogen lysis as part of the secondary response in the event of repeated antigen contact. Macrophage-mediated lysis of the pathogen occurs by binding of the macrophage-resident Fc receptor to the Fc ends of the antibodies on the pathogen. This so-called antibody-dependent cell-mediated cytotoxicity results in the secretion of cytotoxins and bactericides from the macrophages and the destruction of the pathogen membrane. Simultaneously, activated macrophages secrete IL-6 to increase acute phase protein production in the liver. The granulocyte-mediated lysis follows the same antigen-antibody-mediated procedure as in macrophages. The complement system-mediated lysis of the pathogens occurs by binding of C1q to bound antibodies and a conformational change at C1q, which leads to the formation of a C1qRS complex and activation of the complement system via the classical pathway. Ultimately, the destruction of the pathogen membrane occurs via its membrane attack complex, mediated perforation. After pathogen lysis using macrophages, natural killer cells, or the complement system, existing T lymphocytes, Th0 cells, with a specified MHC2 receptor remain in the plasma as Tm cells, T lymphocytes. As part of the secondary response, the Tm cells bind to pathogen antigens in the event of repeated pathogen contact and in this case also proliferate into CD4 Th2 cells, T lymphocytes.
MHC2 also belongs to the antigens of the human leukocyte antigen system. MHC2 plays its major role in antigen presentation of pathogen-specific protein antigens to T lymphocytes, Th0 cells, in the primary response to infection. The MHC2 molecule, which is produced in the antigen-presenting cell, attaches itself to the released pathogen antigens during pathogen lysis and the complex of MHC2 and antigen is turned outwards on the cell membrane. The antigen-presenting cells include dendritic cells, monocytes, macrophages, and B lymphocytes. The MHC2 receptor of the T lymphocytes, Th0 cells, is then modified into a pathogen-specific MHC2 receptor by contact with the MHC2 antigen complex mentioned. Here, MHC2 molecules are transferred to the MHC2 receptor, in which the specific imprint of the pathogen antigen is stamped, so to speak. CD4-Th2 cells, T lymphocytes, bind to the pathogen-specific MHC2 receptors, which allow B lymphocytes to proliferate using IL-5 and IL-4 to form plasma cells and thus initiate antibody production. The major histocompatibility complex, MHC, comprises the antigens MHC1 and MHC2, which are found on the surface of leukocytes and, with the exception of erythrocytes, on most other body cells. Molecules of the major histocompatibility complex are counted simultaneously to the human leukocyte antigen system. MHC antigens are glycoproteins of the IgA class, which are haptotypically encoded by more than 200 genes in the HLA complex on chromosome 6. The MHC and HLA systems ensure chemical individuality and tissue compatibility or tissue incompatibility, e.g. in graft versus host reactions. At the same time, the MHC and HLA systems are responsible for resistance and sensitization to infections, pathogens and diseases as part of the immune response. A deficiency or a change in the MHC complex leads to apoptosis of the affected or modified cell. Secondary response as part of the immune response to an infection. The secondary response to a pathogen antigen occurs through repeated contact between existing antibodies and the known pathogen antigen from the previous primary response. As part of the shift from first contact to second contact with a pathogen, an IL-5-mediated isotype switch from IgM to IgG occurs in the plasma cells. In addition to IL-5, IL-4 secreted by Th1 cells contributes to isotype switching.
Depending on the concentration ratio of various interleukins and other factors, e.g. on MALT-associated lymphocytes, an isotype switch to IgA can also take place. The consequence of the presence of IgG is renewed opsonization and marking of the pathogen via antibody-antigen binding for macrophages, the complement system and granulocytes. The antibody-mediated lysis of the pathogen that occurs follows the same principles as in the primary response. T lymphocytes can also bind to the known pathogen antigens via the TM cells remaining from the primary response and activate B lymphocytes to proliferate to antibody-producing plasma cells by proliferating to Th2 cells and secreting interleukins. Intracellular pathogens and cell aberrations represent a particular target in the course of the immune system's primary response to an infection, since the pathogen-specific antigens are usually not freely accessible on the affected cell surface. Probably the most central role in the detection of infected or modified cells is played by the MHC1 complex, which is changed in the context of an infestation or an aberration. Whereas the unmodified MHC1 complex inhibits lysis mediated by natural killer cells and cytotoxic T lymphocytes, the altered MHC1 complex activates the release of perforins and granzymes from the lysosomes of natural killer cells and cytotoxic T lymphocytes. Due to the perforation by perforins, the granzymes, proteases, can penetrate the affected or changed cell and destroy it. At the same time, the interferons IFN and IFN are secreted from affected cells, which autocrine stop proliferation. Against this background, IFN is used in cytostatic therapy. Macrophages are also involved in the lysis of infected and modified cells. They secrete IL-12, which increases the activation of the natural killer cells and cytotoxic lymphocytes and secretes, as in the classic primary response, cytotoxins that lyse the infected cells and pathogens. Existing Th1 cells activate macrophages using IFN and thus also increase the cytotoxic effect of natural killer cells and cytotoxic T lymphocytes. At the same time, Th1 cells secrete lymphotoxin, which, after perforating the membrane with perforins, penetrates the damaged cell and supports lysis. The ultimate consequence is phagocytosis of the cell debris and pathogens by granulocytes and macrophages. The MHC1 molecule belongs to the main histocompatibility complex as well as to the human leukocyte antigen system and is used in contact with CD8 natural killer cells, CD8 macrophages, and CD8 cytotoxic T lymphocytes, e.g. in the context, the primary response and infection, the inhibition of cell-mediated cytotoxicity. All cells containing a nucleus express MHC1 on their outer membrane and signal their functionality to the aforementioned natural killer cells and cytotoxic T lymphocytes via their MHC1 receptor. When a cell changes into a tumor or when it is infected with intracellular pathogens, the MHC1 complex is modified and, in the case of a pathogen, free pathogen antigens and other unnatural components are incorporated into the MHC1 complex. Natural killer cells, macrophages, and cytotoxic T lymphocytes can bind to this modified MHC1 complex via a second MHC1 receptor and initiate the lysis of the modified cell by releasing the granzymes and perforins from their lysosomes.